Hello students, in previous part we have studied about DNA. DNA was firstly discovered by Friedrich Miescher in 1869. Friedrich Miescher saw the DNA in a pus cell and he called it nuclei. Later in 1889, Altman showed that this is nucleic acid because due to the acidic nature of the nuclei, and the term DNA was coined by Jacaris. We have studied about this one. And then nucleic acid. What are the components of nucleic acid? Nucleic acid. Components of nucleic acid. There are three components of nucleic acid: pentose sugar, nitrogen base, or phosphoric acid. These are the three components of nucleic acid. Pentose sugar. What does it mean? It means it contains 5 carbon molecules. There are 5 carbon molecules. Okay, a compound which contains 5 carbon. Pentose sugar is 2 types. Pentose sugar is 2 types: ribose and deoxyribose. Ribose and deoxyribose. Ribose stand for RNA and deoxyribose stand for DNA. Both, whether it is ribose or deoxyribose, both contain 5 carbon molecules. Then what is the structure of ribose and deoxyribose? 5 carbon molecules in which a ring is produced of 4 carbon molecule and 1 oxygen. 5th carbon remains and form an arm. Ribose, it forms a ring of 1 oxygen molecule and 4 carbon compound in a pentagonal structure. This is the carbon number 1. This one is carbon number 2, carbon number 3, carbon number 4, and 5th carbon remains here. A ring is formed, whether it is ribose or deoxyribose. A ring is formed of 4 carbon molecule and 1 oxygen in a pentagonal structure. 5th carbon remains as free, same as deoxyribose. 4 carbon atoms and 1 oxygen molecules form a ring in pentagonal structure, and 5th carbon remains form an arm. This is the carbon number 1, carbon number 2, carbon number 3, carbon number 4, and this one is carbon number 5. Both are the same structure, but what is the major difference between ribose and deoxyribose? At carbon number 1, it contains H and hydroxyl group, OH. H and OH. H and OH. This contains H, and at carbon number 5, it contains CH2OH. This is the structure of ribose, means RNA. And deoxyribose, why it is known as deoxy? Because in RNA, carbon number 3 and carbon number 2, there are 2 functional group, 2 hydroxyl group. But in deoxyribose, carbon number 1 H and OH. Carbon number 2, here is H and H. Oxygen is reduced. Due to the reduction of oxygen, it is known as deoxy. And carbon number 3, it is H, OH, 4 H, and here is CH2OH. This is the major difference between ribose and deoxyribose at carbon number 2. In ribose, carbon number 2 and 3, it contains 2 functional group. It contains 2 functional group. And at carbon in deoxyribose, there is no hydroxyl group. It contains only H and H. only one functional group, that is carbon number, position number carbon 3. One functional group. So DNA is the genetic material. Why? Because it makes the stability. Why it shows the stability? More functional group compounds can have more functional group more functional group compound is more reactive than the less. Such as ribose. Ribose contains 2 functional groups. It contains excess number of functional groups, so it is more reactive, and more reactive show not stability. And it contains only 1 functional group, so it show the stability. Therefore, DNA is the genetic material, not RNA. We will discuss later. And second one is nitrogen base. Nitrogen base is basically 2 types: purine and pyrimidine. Purine is also 2 types: adenine and guanine. Purine is 2 types and pyrimidine is 3 types: cytosine, thymine and uracil. In case of purine, there is 2 types: adenine, it represented by A, guanine by G, cytosine by C, thymine by T and uracil by U. In case of purine, there is 2 types of adenine and guanine. But in pyrimidine, it is 3 types: cytosine, thymine and uracil. But in nucleic acid, pyrimidine use only 2 types. If it is RNA, then U and C is used, and if it is DNA, then it is a C and T used. In place of thymine in RNA, uracil comes up, or you can say that uracil replaces the thymine in RNA. But in DNA, it contains thymine, not uracil. So use of pyrimidine is 2 types. Only 2 types in nucleic acid, but there are 3 types of pyrimidine. Then major difference between purine and pyrimidine is also purine, pyrimidine. Purine is a dicyclic. It is dicyclic. It means it forms 2 chains. And pyrimidine is monocyclic. Second difference is, in purine, 4 nitrogen atoms. There are 4 nitrogen atoms in purine at position 1st, 3rd, 7th and 9th. And in pyrimidine, there is 2 nitrogen atom. And position 1st and 3rd. Clear? Third one is, this purine attached with pentose sugar at carbon number 1. Means, suppose this is the pentose sugar structure. This is the carbon number 1. Here is the nitrogen base is attached. Purine first at the position of 1st carbon and the 9th atom of nitrogen. And at pentose sugar, carbon number 1, nitrogen base means purine is attached of 9th position. 9th position of nitrogen atom attached with the pentose sugar at carbon number 1. But in pyrimidine, at pentose sugar, at carbon number first, means nitrogen base is also attached on carbon number first, whether it is purine or pyrimidine. But the atom which one is attached here, if it is purine, first position of carbon, ninth position of nitrogen. Or if it is pyrimidine, first position of carbon and first position of the nitrogen atom. At carbon number 1 of first, or you can say that first carbon at first nitrogen atom. Clear? This is the nitrogen base. And next one is phosphoric acid. Phosphoric acid stands for POH, OH, OH and double bond O. It means H3PO4. Phosphoric acid is attached at the carbon number 5th of pentose sugar. Phosphoric acid is attached at carbon number 5th of pentose sugar, and it forms a bond that is called phosphodiester bond. Phosphoric acid is attached at the pentose sugar, means this is the pentose sugar. Carbon number 1, carbon number 2, 3, 4 and 5th here. It is CH2OH. Here is attachment of POH, double bond O, OH and OH. The reduction of H and OH means H2O. Reduction of water molecule, and here is bond formed, and this bond is known as a phosphodiester bond. Phosphodiester bond. The bond which is formed at carbon number 5th due to the attachment of phosphoric acid, that bond is known as phosphodiester bond. And carbon number 1, the nitrogen base is attached. Carbon number 1, the nitrogen base attached, and this bond is formed. Due to the attachment of nitrogen base at carbon number 1, here is also one bond also formed, and that bond is known as glycosidic bond. This is the components of nucleic acid. Then what is the major difference between nucleotide and a nucleoside? Nucleotide. Nucleotide contains pentose sugar plus nitrogen base and plus phosphoric acid. If it is the structure just like as this one, here is nitrogen base attached. This structure is known as nucleotide. What do you understand by nucleotide? It consists pentose sugar, this is the pentose sugar, nitrogen base is attached at carbon number 1, and phosphoric acid attached with the carbon number 5th. This is the structure of nucleotides. And nucleoside means nucleotide minus phosphoric acid. Nucleotide से phosphoric acid reduce कर दीजिए, that is the nucleoside. So we can say that pentose sugar plus nitrogen base. If it is contains pentose sugar or nitrogen base, then that is called nucleoside. And if it is contain pentose sugar plus nitrogen base and phosphoric acid, that makes the nucleotide. Clear? In next part we will discuss on the DNA double helix structures.